embryonic development and patient outcomes. So IVF labs have been designed to provide optimal growth conditions for gametes in culture. Regardless of the environment hardware, there's always software best practices that each lab can employ to ensure an optimal environment. Suboptimal lab environments expose human gametes and embryos to contamination and stress. Bacteria can cause degeneration of embryos, while fungus can retard embryonic development. All of these factors can impact embryo quality, leading to lower pregnancy and live birth rates. Recently, epigenetic impairment due to stressful lab conditions has been described, providing yet another compelling reason to maintain a clean environment. Strategies for maintaining a clean IVF lab include the development and review of protocols, quality assurance of reagents and equipment, quality control of the laboratory environment, and implementing simple checklists for proper maintenance. Each of these strategies can be customized for the end user, providing meaningful methods for optimizing conditions for culture of oocytes, sperm, and embryos. So what classes of contaminants and stressors exist in and around the IVF lab? Well, the main toxicants measured in air are low molecular weight aldehydes, air pollutants, and ozone. Plasticware and equipment may also introduce chemical pollutants through off-gassing, and paper products may introduce particulate matter. Oocytes retrieved through the vaginal wall and sperm delivered to the lab as semen may be sources of bacteria, fungi, and viruses. And similarly, lab personnel are also a common source of bacteria. The four most potent air pollutants are volatile organic compounds known as VOCs, such as aldehydes. Small inorganic molecules such as nitrous oxide, carbon monoxide, substances derived from building materials, again, such as aldehydes from flooring adhesives, other compounds such as aerosols, refrigerants, heavy metals. These pollutants are introduced into the IVF lab most often by overuse of alcohol to clean lab surfaces, thereby generating VOCs, off-gassing lab equipment, off-gassing lab supplies, anesthesia gases, and refrigerants. Particulate matter may be introduced into the lab by paper and cardboard. Air also contains biological pollutants such as bacteria, fungi, and viruses. Culture media may also become a possible source of contamination. Mineral oil or paraffin oil overlay employed during embryo culture provides temperature, pH, and osmolar stability. However, it has been demonstrated that oil may contain mold, zinc, or peroxides that cause sublethal cellular changes. Depending on the storage condition, peroxides may be generated, which may significantly and negatively impact blastocyst development. Therefore, it may be beneficial to wash oil with protein-free medium in order to remove toxins. Chemical and biological toxins may be present or may be introduced by improper culture techniques. Most commercial suppliers of medium supplements, or disposable plasticware must adhere to stringent sterility requirements that greatly reduce the risk of contamination transmission in the lab. Nonetheless, improper storage of IVF supplies or damage during packaging or storage could inadvertently result in contamination. Improper handling of sterile devices, such as allowing the outer wrap of a pipette to touch the sterile pipette, could result in contamination of the culture medium. Attention must also be paid to sterilization methods, as some can produce residual toxicants. For example, a catheter should not be sterilized with ethylene oxide, as this could have deleterious effects on the embryo. All items from manufacturers and clinics should be sterilized properly in order to avoid the introduction of contaminants. Proper procedures for autoclaves and dry sterilizers must be followed, including regular maintenance and monitoring of the effectiveness of these sterilizers using biological methods. Even the products of dead microorganisms, such as the lipopolysaccharide membrane of gram-negative bacteria, can result in the destruction of embryos. For this reason, most fluids and contact materials are tested for endotoxin, and they must pass strict quality control standards. A source of contamination in the lab that can at best be controlled but not eliminated is the female patient who undergoes transvaginal oocyte retrieval and the male patient who provides a seminal ejaculate. All biological fluids should be handled using standard precautions. The most common fungal contaminants in the tissue culture are Aspergillus, which is an aerobic mold, or Candida albicans, which is a yeast. Common bacterial contaminants include E. coli, Mycoplasma, Staphylococcus, and gram-positive rods. Both bacteria and fungi have been identified in IVF cultures. While bacterial contamination often results in the demise of the embryo, fungal contamination identified in the early stages usually do not harm the embryo. In a study by Ben Chitrit et al. And his colleagues in Israel, this is many years ago, 1997, 7 cases of fungal contamination resulted in pregnancies in all 7 patients. Embryos were merely washed free of the fungus and then were transferred to the patient. It may be that when fungi cause harm, it is either via depletion of energy substrates or a buildup of harmful metabolites. It appears that as long as the medium is replaced and the fungal contaminants are removed, the embryo can grow. Peltzer et al. In 2013 examined the follicular fluid of 263 women undergoing oocyte retrieval for the presence of bacteria. They found bacteria in the follicular fluid of all 263 women. 71% of the time, these organisms were also cultured from the vagina, suggesting a vaginal source for the presence in the follicular fluid. Regardless of the source, follicular fluid is not sterile and could be one major source of contamination in the lab. In semen, the prevalence of bacteriospermia has been well documented. Microbial contamination of the semen sample collected for IVF could be of great importance as the natural defense mechanisms are compromised. Although centrifugation through gradient columns will reduce the microbial load of contaminated semen, it will not necessarily reduce it to zero, especially if the microbes bind to the sperm. Bacteria in the sperm fraction can be contained by prophylactic treatment of the male with antibiotics or by sperm preparation in antibiotic-rich medium, which effectively eliminates 95% of the organisms. Foot traffic. Foot traffic in and out of the lab is a significant source of pollutants into the lab environment. And it has been estimated that humans shed roughly 1 billion skin cells daily. Desquamated human skin cells are an important contributor to particles in indoor air. And there is strong evidence that bacteria is associated with these skin cells. Skin shedding may influence indoor air concentrations through both skin cells and their fragments directly becoming airborne, and also by the deposition of cells into the floors and other surfaces, followed by fragmentation and resuspension. Dust consists largely of skin cells and their accompanying bacteria. It has been demonstrated that human occupancy produces a marked increased concentration of respirable particulate matter and bacterial genomes, and bacteria from the human skin and from other environmental sources significantly contribute to indoor bacterial populations. The most common skin flora are Staphylococcus epidermis, Staphylococcus aureus, Acinetobacter, Klebsiella, and other enterobacters such as E. coli. In conclusion, it appears that many typical operations in an IVF lab can contribute to a suboptimal environment. Unpacking cardboard boxes, setting up gas tanks in or near the lab, moving staff or sterile wrapped plastic supplies in and out of a controlled environment, such as an IVF lab, can generate dust, dirt, and contaminants. Given the numerous potential sources for and types of contamination described here and elsewhere, the challenge we face is to identify the best embryo-friendly practices for maintaining a clean IVF lab. Adopting a clean room philosophy is defined by testing and by metrics. So in testing, most tests for IVF lab cleanliness are indirect. Unlike inpatient hospital stays and post-surgery assessments that can measure the length of a stay, readmission rates, outcomes, culture conditions can only be tested by bioassay, which indirectly reflects the cleanliness of the environment. The mouse embryo assay, or MEA, is widely used, though it has been well-documented limitations. Both the MEA and the sperm bioassays measure chemical and biological contamination of the culture system. Contaminants such as endotoxins, which are commonly found in purified water systems, may be specifically tested. Assessing embryo quality at discrete time points within the culturing process can also be a useful way of measuring the quality of the lab operations. Some feel that the quality of embryos derived from donor oocytes provides a control group for assessing optimal lab conditions. Metrics. Several studies describe significantly improved IVF outcome measures after labs have been retrofitted with air filtration controls. Recently, epigenetic and assisted reproductive technologies have been linked, specifically the exposure of early cleavage stage embryos to in vitro culture conditions in an IVF lab. ART involves several steps that subject the gametes and early developing embryos to environmental stresses. And this is the primary reason for an increased interest in the putative link between these techniques and epigenetic imprinting disorders. Although animal studies support such a link between ART and imprinting disorders via altered methylation patterns, data in humans is inconsistent. Current ART protocols employ prolonged in vitro culturing until the blastocyst stage before an embryo transfer, which results in higher pregnancy rates, implantation rates, and reduced multiple gestations. However, extended culture and/or use of different types of culture medium during early embryo development potentially influences the process of remethylation. So in this next part of the talk, I will review broad strategies. This is the third part of the talk for keeping the lab clean. And I know this audience is very experienced and likely more experienced than I am in directing excellent human IVF labs. So I won't delve into basics, details here, which I'm sure are known to everyone here in attendance. So the complexity of an IVF lab must be taken into account when assessing non-conformances. So the American Society, ASRM, has recommended that clinics employ at least 2 embryologists per 150 cycles to maintain a safe environment. Let me say that again, 2 embryologists per 150 cycles. This guideline is being updated, but it remains true that strict quality control and quality assurance, QC and QA, are required to maintain a clean IVF lab. And this is easier with adequate staffing to minimize the risk of shortcuts. Both ASRM and the European Society, ESHRA, have published best practice guidelines for the IVF lab. These guidelines are revised as needed as the field advances and innovations are introduced, but the guidelines address such topics as lab staffing, embryologist certification, lab design, policies and procedures, equipment, and safety measures. They are useful as a reference and a foundation for creating an end user quality assurance and quality management program. that monitors critical indicators of patient care and create an environment that provides consistent, optimal culture conditions. And I encourage all of you to access these documents if you have not already done so. So QC includes all activities or operational techniques within a process. So it addresses both tangible elements, personnel, instruments, equipment, supplies, and intangible elements, techniques, protocols, documentation, record keeping. The main goal of QC is to evaluate effectiveness of policies and procedures and identifying correct problems, assure accuracy and precision of procedures, and monitor performance and competency of the lab staff. Quality assurance provides a systematic monitoring of the testing process in order to help identify problems, errors, or improvements that may have occurred. Results should be evaluated on a regular basis. Indicators should be objective and relevant, and adequate thresholds should be established. Critical levels of lab performance for each indicator should be defined. While these indicators evaluate the entire process in the lab, they also indirectly reflect the ability to provide optimal culture conditions that are predicated on maintaining a clean IVF lab. Assessing QA in the IVF lab presents certain difficulties. The final anticipated outcome is an ongoing pregnancy, which entails numerous components that are beyond the control of the lab. The following key performance indicators, however, can be regularly reviewed, analyzed, and discussed. Rates of normally fertilized oocytes, cleavage rates, rates of embryos of good quality, blastocyst conversion rates, proportion of patients with failed fertilization, ongoing clinical pregnancy rates, and implantation rates. Unfortunately, identifying a true decline in performance often occurs several weeks later when pregnancy rates decline, delaying the implementation of corrective action. Therefore, it is essential to establish minimal risk strategies in the form of strict discipline of routines and procedures, ensuring QC and QA. Attention to quality is a dynamic process to be clearly defined and documented, with general policies and delegation of responsibilities carried out with continuous monitoring. So in the next few slides, I'll just discuss some strategies on how to keep the lab clean. And this slide, we talk mostly about controlling lab air. So two of the most common contaminants in the lab air are VOCs, volatile organic compounds, such as aldehydes, and biopollutants. Therefore, the lab should have a dedicated and specialized air handling system with the following elements: positive pressure, high filtration, high frequency of air exchanges, and climate control, namely temperature and humidity. The air handling system should function according to set parameters, and periodic testing for VOCs in the lab air should be implemented. Air filtration systems should ideally be installed at the time of lab construction. Less ideally, labs may be retrofitted and/or floor units containing fans and an air filter may be placed in the lab. Most modern-day labs have installed into their air systems some or all of the following filters: high efficiency particulate absorption, which is HEPA filters, carbon filters, or carbon potassium permanganate filters. Additionally, the following modifications may be made to the environment in order to help control air contamination: frequent hand washing and glove changes between patients. Urge embryologists to wash their hands with biological soap and tap water each time they enter the lab. The safety data sheets of the biological soap should be reviewed, and only soap without known hazardous ingredients should be used. Not all soaps have been evaluated for reproductive toxicity, unfortunately. Use high quality distilled water in the lab. This should be high purity, non-pyrogenic, endotoxin tested, possibly MEA tested, commercially available for embryo culture. This can be used to clean surfaces and to dilute any cleansers you may be using, such as hydrogen peroxide. Equip the IVF lab, if possible, with laminar flow hoods. These confined workspaces with stable vertical unidirectional airflow, which is laminar, provide protection against contamination of items in the hood. Laminar flow hoods may also have HEPA filters and should be turned on at full high speed each morning at least 10 minutes before the start of procedures, unless you leave the fans on all the time. And this is in order to recirculate the air, fresh air through the filter. Reduce or ideally eliminate the use of 70% ethyl alcohol or ethanol when cleaning surfaces. The level of alcohol measured varies from lab to lab. Alcohol used to wipe down surfaces becomes airborne, introducing VOCs into the lab air, which can deposit into your air filtration system. Instead, clean lab surfaces regularly with distilled water and decontaminate with hydrogen peroxide, either at 3% or 6% concentration. Only distilled water should be used routinely with a lint-free wipe to clean surfaces of hoods, counters, and heating stages. For decontamination of surfaces, including the hood between oocyte retrievals and after the last retrieval, use 6% hydrogen peroxide on a lint-free wipe followed by a rinse with distilled water. One can also use 3% hydrogen peroxide or other low VOC cleansers on your walls and on the floors of the lab. Incubators today are largely benchtop, either dry incubation or with a small water reservoir providing humidified culture conditions. Incubator QC and QA usually follows manufacturer's recommendations. Here we discuss how to control the physical plant and the staff. Well, we must ensure that the IVF lab is protected and secured. It should be protected from potentially toxic chemicals such as cleaning supplies, detergents, fragrances, and fumes as much as possible. Restrict access to the lab and the procedure room. Service technicians and other authorized visitors should always be accompanied. Require embryologists and other authorized personnel to always wear scrubs, masks, and head and shoe covers. Insist that embryologists are attentive to sterile technique. Skin cells and bacteria can exfoliate while reaching over dishes and cause inadvertent contamination. Confirm that no painting, renovation, repair is being done around the lab, and ensure that noise and vibration levels are acceptable. Here we talk about controlling protocols and workflow in an effort to keep the lab clean. These should be reviewed to ensure efficiency of all lab and clinical procedures. Protocols should require attention to detail and to sterile technique at all time. Maintenance of a clean IVF lab requires optimization of the QA and QC protocols in particular. The following is a list of possible QC and QA protocols that may be used to create a personalized QM program. So you can document all QC, QA, and corrective actions in order to provide transparency and traceability. Create a program to reduce or eliminate hazardous materials such as mercury thermometers, sterilizing products that contain ethylene oxide, betadine, or are otherwise embryotoxic. Schedule and monitor equipment, QC and QA. Tolerance limits on equipment should be observed at all times in order to maintain optimal culture conditions. The physical condition of equipment should be monitored in order to avoid breakdown, overuse, or expiry that can introduce contaminants into the lab environment. For example, overuse of gas tanks. So if you have a gas tank where the PSI is down below 400, this can stir up contaminants from the bottom of the tank into the lab supply. So in my labs, we change our tanks always at 400 PSI minimum. In addition, incubator seals can break down from overheating. Dust buildup can occur in equipment that's not routinely in use, and degradation of tubing can introduce contaminants into the gas supply. Regularly scheduling equipment QA for laminar flow hoods, incubators, and microscopes ensures proper operation and cleanliness. All equipment should undergo an electrical safety check annually. Service the air filtration system at least annually, and if possible, do a testing and balancing of the entire HVAC system every 2 years. The following modifications can be made in order to help control what enters the lab. Movement into the lab should be restricted. Only staff that needs to be in the lab should be permitted to enter, appropriately attired in scrubs, mask, hat, and shoe covers. Supplies should be unpacked outside the lab area in order not to bring in cardboard and increase air particulate count. Supplies that are sterile and wrapped in plastic should be stored in cabinets and drawers to minimize introduction of off-gassing compounds into the air. All new equipment should be off-gassed prior to replacement into the lab in order to minimize VOC content in the environment. Biological soap and water should be used routinely to wash hands prior to entering the lab. If a new cleaning product is introduced, it must be reviewed and approved prior to use. Areas should be designated as clean and require glove use at all times. Other areas should be designated as dirty, where items such as paperwork, computer work, cell phone use, etc., occur. As we live in a society where cell phones go everywhere, and we may even send and receive texts from clinical staff members during the day, it should be wiped clean before entry into the lab and used only during restricted times. A cell phone or tablet that is routinely used outside of the lab is one of the most highly contaminated items we own. The following modifications can be made to an environment to help control what is removed from the lab. Obviously, biological waste should be removed daily, if possible, after every retrieval, and wash hands after removal and installing a new bag. Regular waste should be removed as needed from the lab. This is very important, particularly in the context of the global pandemic. There should be no hand sanitizer in the lab, which is very high in alcohol content and therefore VOC content. This will definitely impact your filtration system. So the final part of this discussion centers around creating a culture among lab staff. The most important way to ensure a clean lab environment is to create an atmosphere of discipline, effective communication, and general attention to detail among the staff. The lab should have clear and documented QC and QA programs that are implemented daily and officially reviewed annually by all staff. Regular staff meetings can help with clarifying protocols, setting goals, assigning responsibility, and fostering a team environment through open communication. Lab maintenance and cleanliness can be reviewed periodically, with emphasis given to the areas that need remediation. In the Checklist Manifesto, published in 2009, Dr. Atul Gawande describes the valuable and critical use of checklists in a number of professions. Using his own research, he designed a study to apply checklists to medicine, and specifically to surgery, the findings of which were published in the New England Journal of Medicine. Dr. Gawande and his co-authors concluded that implementation of a checklist was associated with a concomitant reduction in the rates of death and complications among patients at least 16 years of age who were undergoing non-cardiac surgery in a diverse group of hospitals. There are very valuable lessons to be learned here. Checklists, as Dr. Gawande demonstrates, provide a kind of cognitive net, catching mental errors of memory, attention, and thoroughness. They allow for tracking and for communication, and it can make reliable management of maintaining a clean lab very routine. Checklists supply a set of checks to ensure that a routine but critical items are not overlooked. They also supply another set of checks to ensure that people talk and coordinate and accept responsibility while maintaining the power to manage nuances. Good checklists are precise, efficient, and easy to use in the most difficult of situations. They are not meant to be comprehensive, but instead provide a reminder of only the most critical and important steps in any given process. Above all, checklists are practical. When making a checklist, there are a number of key factors to consider. First of all, define a clear pause point at which the checklist is supposed to be used. This might be at the moment before you start work, at the beginning of the day, at the end of the day, when you close the lab for the night. A pause point should be repeated daily at the same time so that it becomes routine. Second, decide if you want a do confirm checklist versus a read do checklist. So in a do confirm checklist, team members perform or do their job from memory or experience. Then they pause to run the checklist and confirm that everything that was supposed to be done was indeed performed. With a read do checklist, tasks are carried out as they are performed. As they are checked off. It is important to pick the type of checklist that makes the most sense for maintaining a clean IVF lab. The rule of thumb is 5 to 9 items total on the checklist, with simple and exact wording that fits on one page and is free of clutter and unnecessary color. Just checking boxes is not the ultimate goal of a checklist. Embracing a culture of teamwork and discipline is. The checklist is a reflection of our code of conduct as individuals and as a team working in the lab. Selflessness and expectation of skill and expectation of trustworthiness and discipline. So here's an example of a possible do confirm checklist for the IVF lab, likely for the beginning of the day and the end of day responsibilities. The main categories of the daily QC are addressed. There's only 5 listed, and the embryologist here would do each task and then confirm with the checklist that those tasks were done. Audits. Audits are a good part of a good QA and quality management program. It is the review of prescribed procedures, as you know, often in the form of checking standard operating procedures and rearranging the procedures if required. A system that monitors individual performance of team members with regular appraisal is also helpful in maintaining an optimal standard of results. Measurement and feedback are crucial elements in defining quality management, which means that assessments and audits, both internal and external, should be applied systematically and periodically. The result is increased traceability and transparency, which helps define the best practices that a lab establishes to maintain a clean environment. So in conclusion, maintaining a clean lab is the first and last step in providing optimal embryology environment for the patient. Many engineering controls are used in the design and construction to ensure that lab access and air quality are controlled. Quality control and quality assurance programs are established and utilized on an ongoing basis to ensure that best practices are followed and that the measures of embryology success are appropriate. Finally, embryologists, individually and collectively as a team, must create a culture of discipline and attention to detail in order to maintain the lab daily. Simple checklists may be helpful in ensuring that basic tasks are completed, thus enabling the embryologist to focus on the art and science of embryology. Thank you very much.
Thank you very much, Dr. Sangeeta, for that awesome presentation.
Most welcome.
Air quality in an IVF lab is like a phantom in the room. It is something which you can't see, but then it also wouldn't let you stay in peace. Until the Cairo consensus came in, many of us didn't even know what to look for in air quality to optimize air quality in the lab. What are the parameters which we have to check, and what would be the values to deem the air quality as optimal or not? So today, you have very lucidly presented the factors, the causative factors for poor air quality in an IVF lab, and what is that we have to look for, and the strategies to improve air quality in the lab. I'm very much impressed with the practical points which you have reiterated. like washing oil with protein-free medium, and like use of restricting the use of cell phones in the laboratory, and the importance of checklist. I think every slide had an important point to take as a take-home message. Thank you very much. So before we move on to the next session, I think we can take 2 questions. Can we?
Yes, please go ahead.
Okay. So
I'll
I'm
stop
just
sharing.
okay.
There we go.
And I'm taking the liberty of asking the first question. So you said, like in your lab, you would change the CO2 cylinders when the pressure is down to 400 psi. So do you think a Coda filter would help if you just let the cylinder run to the last bit?
So we do have gas line inline filters. We do have those. Life Air out with those. I don't think they're available yet commercially. So we use the ones from, I think from Global, or it comes from Cooper now, I suppose. And these are inline filters, but we feel that below 400 PSI, the contaminants in the bottom of the tank, you know, these tanks are recycled. It's not medical grade gas. That's so costly, so I don't use that. We just use common sense, and we just stop at 400 PSI and change the tank. So they are filtered. The gas coming into the incubators is filtered that way, but we still are very, very risk averse, and we change it at 400. And that's our mixed gases, which go into our incubators, and also the CO2, which we just have one incubator, that's CO2. It's a culturing incubator.
Oh, that's very interesting. I think, you know, like many of us would take this point very seriously and start implementing in our laboratories. We have the second question from Raisa from Indonesia. She wants to know if the sperm preparation room has to be a clean room, because literature says that VOCs can affect fragmentation, DNA fragmentation of sperm. And she is asking if a clean room would decrease DNA fragmentation levels.
So the first part of that question, I would say you don't need a clean room. I have labs, my lab in Miami, they don't even have a laminar flow hood. They prepare the semen samples for IVF and IUI together in the andrology lab. They pass them to the embryology lab. The embryo lab is under Life Air, and that's where they're doing all the semen ICSIs. So, but the processing does not happen in a clean room or even in a hood. And that has not shown an impairment that we have detected in terms of embryo development or sperm quality. Now, having said that, you know, the sperm, I feel so badly because the sperm is often overlooked in favor of the oocyte. And I do prefer that it is processed in a laminar flow hood. I keep the centrifugation speeds low so that I don't introduce reactive oxygen species from very fast spinning. And one more thing I wanted to mention, which was not in my slides, especially in the context of COVID, but this came up also in the context of Zika virus, and we do infectious patients as well, Hep C and HIV. We use a double tube method to prepare the sperm, and I'm sure you're familiar with that. It really gives you a very, very clean final pellet for therapeutic use, and this is a double tube method that really removes the bulk of any viruses that may be in the semen.
Okay, thank you very much. I think we have one last question. This is from Drishti Srivatsava, and she wants to know the correct way to use hydrogen peroxide in the embryology lab. Can we use it on the lab floor?
Yeah, so hydrogen peroxide is sold commercially at 3% concentration. So that's the easiest way to use it, and you can definitely use that. In the era of COVID, we've actually changed our policy. We're now spraying directly onto the surface of a floor or a counter or a laminar flow hood, which is metal, stainless steel, and we allow it to dry and thereby, you know, kill the viruses of any kind that may be there. And then we follow with a wipe of distilled water with a lint-free cloth. The other way you can do it is to buy 30% hydrogen peroxide, which is sold from, you know, Fisher Scientific, for example, and you can dilute that with distilled water to 6%. So 6% is actually what I use on my counters, and 3% is what I use on my floors.
Okay, thank you very much. Ved, do we have time for one more question?
Yes, yes, please go ahead. There is no problem. We can take.
Okay, this is from Dr. Karan, and he is asking Dr. Sangeeta's thoughts on current COVID infection control in IVF labs.
Yeah, so everyone's asking that. And I must confess, I think the lab is the safest, cleanest place to be honest. We have, I mean, my labs, I know they have very highly engineered air filtration systems, positive pressure, very clean filters. We change the filters actually more often, I would say, during COVID, just to ensure that there's a safety comfort level that everybody has. I've definitely restricted access to the laboratory, and we have just augmented our cleaning protocols. We don't share pens. We have teams of people, so not everybody's in the lab at the same time, so that if somebody has to go out and quarantine, I'm not left without staff, because everybody then has been exposed. But I think if you follow augmented QC and your laboratory hardware is in place, I think there's no problem. With our patients, we are screening our female patients several times during their cycle. They must not be positive for COVID by RT-PCR. I will confess, though, we're not screening our men. And so they do not produce their samples in the office. They must bring it from home, and they must wear masks and, all of that stuff that we take precautions, social distancing. So we have altered that and have reduced the amount of traffic in the office and the access to patients. So there's good things and bad things about that. But the laboratory itself, just augmented cleaning and common sense.
Thank you very much, Dr. Sangeeta. I think now we can move on to the next session. Thank you very much, IHERA team. Yeah.
Thank you. Thank you, Dr. Sangeeta. Thank you so much. Thank you, Dr. Sujata. And I think we can move to another second session. Sanket?
Yes, sir.
Please start.
Dr. Rahul, can I have the slides, please?
Just a second, Sangeeta.
Sure. Thank you so much, Sangeeta ma'am, for the wonderful lecture. So as Sujata ma'am said, we all had at least one take-home message from every important practical point which you emphasized on. Thank you so much.
My pleasure.
So moving forward, we'll be starting our panel discussion. So I'll be introducing moderators for today's session, Dr. Keshav and Dr. Gaurav. So Dr. Keshav holds a master's degree in clinical embryology and one of the few STA certified clinical embryologists in India, and is the lab director for Rainbow IVF and now the director for META, Malhotra Embryology Training Academy. And he's on board with ISAR and ISPAT, and has worked on the key projects pertaining the embryology in the country, like occupational standard for embryologists and the embryology consensus. So he was also awarded with Budding Embryologist of the Year 2019 by Economic Times. And the next moderator is Dr. Gaurav Majumdar, who is working as a Chief Embryologist at Center for Reproductive Medicine, Sir Gangaram Hospital, New Delhi. He has earned his MSc in Clinical Embryology from National University of Singapore in 2001. And he received his PhD in research work on PGS for aneuploidy. And currently is a director for the center at Sir Gangaram Hospital, which performs around 1,200 IVF or XC cycles and 750 frozen ET cycles and annually. And 2014 is successfully set up a PGS PGT program, which now successfully runs in-house genetic testing facility using qPCR to screen embryos for aneuploidy. And he was also awarded for the Best Embryologist of the Year India 2019 by Economic Times. And he has numerous publications in international journals, has contributed 4 book chapters in ERT textbooks. He is also joint treasurer for Academy of Clinical Embryologists India and executive committee member for Indian Fertility Society India. So without taking much time, over to both.
Thank you, Sanket. for the introduction. And so, Keshav, should we start?
Yeah, looking forward to it. And looking forward to actually having a lot of interactions with the panelists. Sanket, thank you for that introduction. And I think Dr. Sangeeta has already covered most of the points that we are wanting to discuss today. But what we're trying to achieve from this panel is to have like a more practical experience in setting up IVF units, how to actually handle air handling units, which one to select, what are the challenges that can happen with air handling units, as well as with air quality in the laboratory. So both hopefully like both me and Gaurav will learn a lot from this panel is what we are intending to do today. And without further ado, I think Gaurav, the floor is all yours.
So just at the outset, I'd like to make a small confession that I am actually a skeptic when it comes to air quality. So I am going to be playing the devil's advocate to this panel. And it's a very interesting topic. It's been very cleverly worded that air quality management, how much is too often, is too much. So it actually automatically, subconsciously makes one feel that, okay, probably it is required, but we are still not sure to what extent. And it is still, through this panel, we'll try to address this question that what is the true value of air quality management, and where does it stand in the scheme of ART, and to what extent, that beyond what point is it, it's not worth investing in it. So we have a nice panel of experts who are going to be with us in this discussion. So I will just read out the names of all the panelists. So we have Dr. Alex Verghese, who is the co-founder for Life in Vitro, scientific director of the Astra Fertility Group. Then we have Dr. Ashish Sood, who is the scientific lab director for Neelkant IVF. And then we have Dr. Vijay Mangoli, who's the laboratory director for the Fertility Clinic and IVF Center at Mumbai. Then we are also joined by Mr. Maju Jacob, who is the COO of Kraft Hospitals. And finally, last but not the least, Mr. Upendra Sathe, who's a chemical engineering professional, joining us from Pune, Maharashtra.
I can't see Ashish and Upendra. So if you can please switch on your videos.
Sorry, Keshav, actually Dr. Ashish is not going to join us because there is some problem in his family. So he's not going to come.
And Upendra Sathe?
Upendra sir is there.
Perfect.
Good evening.
So we have a very wide panel today. We have like clinical directors, we have COOs, we have an engineer amongst us. So we're looking forward to a fantastic discussion.
Because we have Dr. Sangeeta also.
Yes.
I just saw her and I'm so glad that she's hanging around for this. So we are going to get some inputs from her as well, I hope.
I'll hang around until I have to go to the lab and check fertilization.
Sure.
Right. So I think air quality, as Dr. Sangeeta has said, is one of the prime determinants of reproductive outcomes, which is often missed. Now, Richard Legros in 2010 actually noted the effects of declining air quality on reproductive outcomes. And it's not just the VOCs, it's also the particulate matter, it's also the contaminants, etc., which all of us have to be really bothered about. So Gaurav, would you like to take the first question and put it to our panelists?
All right. So this is a very pertinent to the panel here, and where especially for all the people here who are attending, they'd like to know that if, and I'd ask this, I'm posing this question to the entire panel, is there actually enough published evidence investigating the effect of air quality in the IVF laboratories on pregnancy outcome and embryology outcome. And because this evidence is critical in order to believe in the air quality management system. So Dr. Vijay and Dr. Alex, you could just make a start here.
Sure. I think Gaurav, if you start looking for in the literature and publications, more than 3,000 papers have published in various journals, on the effect of the air quality on various cultural aspects. It includes the outdoor air, indoor air quality, the air pollutants they are generated from the disposables due to activities like disinfectants, cleaning, off-gassing of it, as mentioned by Dr. Jindal very aptly. Plastic waste, the ozone production, UV radiations, air contaminants from the anesthetic gases and other activities, OT activities. There are various aspects which are published, and most observations they conclude that the polluted air has a negative effect on the IVF outcome at certain points of time, various points of time. Not that all the pollutants have the same effect and same intensity effect on, including the fertilization, including the cleavage, including the blastocyst formations, or including the pregnancy rates. But various different aspects have studied into this. Now, VOC, if you see, the most common factor that is found, it is any organic compound, the carbon containing molecule that gets converted into the gas at room temperature. And generally TVOC, total VOC, is the total concentrations of the combined VOCs which are present, which is exclusively studied. It is logical that they may be having an adverse effect on the embryonic growth, because what we have to remember is the embryos don't have any protective mechanisms like the adult cells. They don't have the epithelial layers, they don't have the immunological defense systems, they don't have any detoxifying mechanisms to separate and destroy the toxins. Secondly, important point is that the air pollutant, these kinds of pollutants, mostly they have the hydrophilic nature. So they immediately, once they are present in your air, in your culture room, they immediately enter inside the culture medium, and they come directly in contact with the embryo. So these are the factors which are published.
So Dr. Vijay, but just building on what you said, that it seems logical that air quality will have an effect on embryological outcomes. But Alex, I'd like to now ask you here that even though it seems logical and there's an impression that air quality does affect, and in fact, I'm surprised to know that there have been 3,000 articles which have been published in this regard, but still air quality is the most neglected aspect in an IVF laboratory. So, because I feel as an individual that there are so many different variables which are there in an IVF laboratory, is it actually even possible to assess the true role of air quality on embryological or IVF outcomes? So Alex, according to you, what would be a perfect study, or what form of evidence or what would be the ideal study for us to actually, understand the contribution of air quality towards an IVF outcome?
Sure. See, if you look at most of the studies that came out, these are observational studies.
Yeah.
And since we are using the human IVF, you can't do any prospective studies in this kind of scenario. So you have to depend on the retrospective nature of the study. And I should say one of the best evidence came from Dr. Amy Sparks' lab in University of Iowa. That is one of the best evidence.
Okay.
Maybe some of you might be knowing about that incident. I think in between, the biomedical engineers removed the carbon filters without knowledge of the lab director.
Okay.
And they analyzed the blast conversion rate, pregnancy rate, and the cleavage rate before the carbon, during the carbon absence, and after a replenishment or a replacement of the carbon. And they have seen a dip in the pregnancy and blast conversion during that time when the carbon was not there. So that's a clear evidence so far, what we have seen in many of the papers. I think these kind of evidences. And the second, the observational study, I think in the last 5 years, we are seeing a lot of observational study when people move from a old style laboratory to a new purpose-built laboratory. Then you are seeing the increase in the implantation rate and live birth rate. I think these are the best evidences. Even though there are some caveat, maybe they must be using maybe some different system embryo culture, but I think as so far, I think weightage is there towards the quality of the air and how the new systems are being implemented.
Okay, so that's very interesting. That what you were suggesting is that the best evidence which came out on air quality was a result of an unknown.
Yes,
it's an accidental evidence.
Accidental, yes.
Accidental evidence.
Anecdotal, yes.
Yeah.
All right.
Gaurav, let's bring in Jacob here and ask him whether, like, as COO, how much importance does he pay? Like, how much importance does he pay on air quality amongst his laboratories at Kraft?
Yeah.
Yeah, thank you, everybody. Thank you, Alex, sir. We together working for a new lab for the last 18 months. And today I have actually commissioned my lab and we are getting our parameters in the proper fashion what we want. So here what I want to highlight is, it's not as a CEO of Kraft Hospital, but I am coming from a cross-industry perspective, because I am coming from life sciences industry, whereas in my role in the university, I was been managing life sciences production and research center in NMIMS Institute, where we used to work on stem cells and other cells which we were researching on developing new clones. So there we use air filter system. Here we use air filter system. So I had a lot of opportunity to learn how I can help an embryology team in achieving their ambitions. So it's their wish list. So first taking a wish list from the embryologist is very, very important before we start working on what we can do. Second is solid background of fluid dynamics and environmental engineering is a requirement for designing any of the labs. So what happens is when any commercial organization engages a third-party contractor or a designer, what happens is he basically comes from HVAC background. It's a high volume air control background. The problem is HVAC basically deals with humidity and it deals with the positive pressure, and it deals with, it doesn't deal with much of filtration. So because they need to bring the particle size only. They don't need to bring all other elements which you are talking in volatile organic components and other components. So that's one challenge where the industry, which is your supporting industry, is not understanding actual requirement of yours. And they're perceiving you as a healthcare institution, not as a biological institution. So here, what my contribution or my talk with Dr. Alex has given me a lot of insight where I can work on several things is because I understood that this particular unit in a hospital is not related to healthcare. It's related to biologicals and it's related to lab practices. So we worked from that angle. So several things which, one instance which I can say, while you construct, so the sequence which you need to go, whether I gone through that all sequence, that I cannot tell you, but in this process, I understood that the sequence first you need to understand is you need to study your environment before you start designing. First, study the environment, then studies your particular infrastructure. Then you start designing it. While you design, you have to think about all future eventualities. Tomorrow, you can have a sudden off-gassing inside your lab or a sudden infection in your lab. How you should keep a route out to clear it off. So that's another design component it has to come.
Yeah, we'll come to the design components, Mr. Jacob, in a while,
right.
But I think very valid points that you have to study the external environment, you have to study the internal environment, and then only try and design a laboratory. And it's great to know that CEOs are actually asking embryologists for wish lists. I think that's something that's a very welcome change. And kudos to Alexa for actually initiating like this dialogue with CEOs. Let's move ahead because we have a lot of questions and there's so much to be discussed. Now we know that inside out, the indoor air can be like 10 times more polluted than the outside air. But this scenario changes in a country like India, in a country like, in a city like Delhi, in a city like Agra, where I also come from, because like the air quality, like Gaurav, have you checked your weather app in the last year or so? And have you seen like even the air quality coming into the orange category in 1 year? Because in Agra, that has not happened.
And then already now, this time of the year, it's rapidly deteriorating air quality. But I wish there was some way of quantifying the effect of this air quality on our outcomes.
Yeah, so how important is it to take into consideration the air quality of your city? And like how important is it in your IVF lab designing, etc.? So I think Alex sir can take it, and we'll take comments from Dr. Vijay and even Dr. Sangeeta at this point. So you're muted, you have to unmute.
Yeah, definitely. Like the cities, we know that air pollution is very high, even though we are seeing a considerable less pollution after the pandemic, maybe in the coming last few months. But it's a fact that most of the metro cities where the IVF labs resides is contaminated with PM10, PM2.5, SO2, and nitric oxide and ozones. And in one of the slides that you have shown previously from Chinese study, I think they studied approximately 1 lakh live birth or something like that. And they have correlated the nitric oxide and ozone in the laboratory environment with the FET outcome, not with the fresh blastocyst outcome. So I think that's a real fact. I think so. We have to be careful when we design the laboratory in a proper way with a proper particle count reduction and also the VOC filtration. Yeah.
I think there's just one paper which has come from Delhi where they've compared the air quality of Delhi, and it's in the months of October, November, and December that they saw a decline in their pregnancy rates and blastulation rates, and the rest of the year it was okay. Vijay sir, your comments on such research papers?
Sure. Before that, I would like to just comment on one thing, that whenever anybody wants to start any IVF center, they have to consider, this is one of the primary considerations that they have to do, is the location of their IVF center where they want to, get up. For example, even if they want to set up their IVF center at the periphery of a growing city, down the line, they have to think about after 5 years, because after 5 years, they may end up into being a city center of the city. So these are the things that have to be considered. Second thing is also they have to study the future projects that are coming up near that particular area, whether any petrol pumps are allotted nearby, whether any industrial area is coming inside, whether huge complexes, whether either industrial or the residential are allotted, they are coming up. So these are the factors they have to consider, because otherwise it will be very taxing on the air purification systems as such to get a cleaner air. Now you can imagine
by that logic, then no center can open in Delhi and Bombay.
No center can last in Delhi. That's true. By that logic.
Any Indian metro. So at the end, your air purification system is what will save you.
Right.
Yeah.
I told you. But the thing is, it will be taxing if you know that such things are coming. But it is true that you have to keep an eye at, as Dr. Jindal has rightly mentioned, that it depends upon how then frequently you have to keep on changing your air filters and the maintenance part of it. That is that will play an important role.
I'll bring in Dr. Jindal here because let's use her till she is here. You're from a city which has relatively good air quality, although in the middle of the city, you can have a lot of pollution with the traffic, but it's relatively great. I was in New York last year. And like, how frequently do you think that you're changing your filters? Is like, does the pollution determine that? And like your aspects, like your comments on this aspect?
So yeah, this is very good. Several points. So I have a laboratory in California, which does
Forest
not have
fires.
for exactly. So we had to change our HEPA filters 3 times in 12 months, which is not cheap. But I insisted because I don't know that they were not exhausted by the fumes. And I didn't want to find out the hard way, the way poor Amy found out when her blast conversion rate dropped like a stone. So that is not a life air system, but we have HEPA filters, we have carbon filters, and we have floor filters in the laboratory. My lab in New York City has a hard ceiling with access panels. So this very much protects the laboratory and keeps the environment, if it's clean, it keeps it cleaner longer. It doesn't have, how do you say, panels which would have grids and, you know, the loose tiles. It's a hard ceiling with access panels. There's no life air there, and we change HEPAs once a year, and we change carbon filters twice a year. My life air filters, I have 2 labs that have life air, and you know, they have this additional feature of ultraviolet light built into the system, which is very proprietary, of course, but it's very interesting that they do that. And I think that's why their system works so well, but it's not cheap. And I can't say that it's available to everybody, but that we have definitely seen a huge increase in blast conversion rate in my life air labs.
Yeah. And I think like you have photovoltaic systems which can also be incorporated into the HVAC, which was quite a like marketing selling point during the pandemic as well, especially during the times when WHO said that, yes, this is airborne and all of those things happened. So these are also things that I think all of us should know about.
All right.
Gaurav.
Yeah. So just one quick question to Dr. Sangeeta. So if I understood correctly, some of your laboratories have the life air system, whereas one of them does not have the air purification system.
Oh, three out of the 5 do not. 2 of them have.
Okay. And do you see any difference in the KPIs or the outcomes of those laboratories?
Right. So I built a QA dashboard, which is has macros. It's an Excel file, and I'm able to get everybody to send me their data monthly. And I look at the KPIs. I use the Vienna consensus. There's 13 KPIs, and I look at maybe 4 every month. And what I'm seeing is that the life air labs, their technology, each culture system is slightly different. The media, whether they change media, it's one step. The patient demographics are different, you can say. But in general, the blast conversion rate is the only metric that I've noticed. The FERT rates are the same. Pregnancy rates actually are fine. Everybody does very well because we're all doing freeze-alls and optimized uterine environment for FET. In general, that's the policies we're following. So everyone is doing pretty well, but the blast conversion rate is much higher in my life air labs.
Okay, that's heartening to know. We'll hold on to that thought. So, okay, so let's come to the next question. So what are the hurdles faced by the IVF units in getting the right clean room construction firms, specialized to or custom made for IVF? Any comments by the panel?
Yeah, see, that's where the whole, because I think Dr. Alex has been put me into this panel because of this particular challenge which we faced in the last 1 year to build a lab. The biggest concern is that three important things, and I quickly want to go to it. One is that the normal contractors which is available to you and the consultant which is available to you is an HVAC consultant. Mm-hmm. And the IVF industry is not choosing a consultant from the biological life sciences industry. 2, you don't have a consultant who have a research opportunity into the environmental components and other recommendations which is required to build the filtration process. So in this case, what available with me was one is Camphil, which been able to give me some support on the filtration side, which to do some research. On the airflow side, we had our internal capabilities, and I used Fluid Dynamics Research Center people from the university, NMIMS University, to get the fluid dynamics and HVAC pressure controls. So what you require is that there should be a team of engineering people, designers should come out in India. I'm not talking about outside of India, who basically have the life sciences background, who has worked on the life sciences like Serum Institute in the vaccine lab, Bharat Vaccines, or Venki's vaccine labs, and that type of labs, or like Reliance Life Sciences, or that type of.
Okay, so Mr. Jacob, so these guys who you're saying who have a life sciences background, they will be able to do a better job as compared to maybe other guys who are also clean room specialists. So they will be able to cater to our needs in a better fashion. That's what you mean?
It's not them alone. You need an environmental specialist. That's very important in your team. While you decide, you need an environmental specialist. Second, you need to have a molecular filtration specialist. Third, you need to have a HVAC specialist. These three components is required. It is not the commercial specialist. It's the technical advisory specialist. Okay. Then you need to have a designer, which is normally available to you, all of you, from any Blue Star, any HVAC company in India. You will get a designer. But on the back, you need these three people. Basically, this is available not openly in the market. You need to either pull it out from a university or from a life sciences company.
That's
I want to commercial. Gaurav, I want to bring in the audience here and just a quick yes and no Q question, whether how many people have actually done this in their laboratories. Just type in a yes or no and we'll try and get some feedback from the audience.
Okay.
Yeah.
Okay. All right. So that was a great point that we need alongside the engineers who are building the clean room. It's important to get biologists also into the whole scheme. Okay. Let's move to the next question. Keshav. Dr. Sangeeta, would you like to make a comment?
No, I'd like to say goodbye.
Right.
Thank you so much for.
Thank you.
It's a pleasure.
Thank you so much.
Thank you.
Thank you so much, Dr. Sangeeta. Thank you so much.
Okay, so we have another clinical.
See you again.
We have a clinical question now, and this basically is from a laboratory which says that they have a dedicated AHU, but their outcomes are not at par with the big centers that are there in the same city. Now, what apart from the air handling unit is needed, and what could have gone wrong in such a laboratory? It's a very open-ended question. I can take your comments.
Short answer. Very, very short answer.
Yeah. But from we can involve all the panelists with a brief answer.
Can I start?
Yeah.
Yes.
Yeah. So in most of the clinic who want to go with the HVAC system or AHU system, they usually approach the company who is making the HU for the pharmaceutical industry or the operation theaters. So their concern is to bring down the particle count. And we can see many of the Indian IVF labs are made in that fashion to reduce only the particle count. And they have maybe the same HU functioning for both the IVF laboratory and the OT, where the egg recovery happens. And you won't find any kind of activated carbon or VOC filters in the system, because this common, the clean room engineers are not aware of, because they don't deal normally in the pharmaceutical or other industries with that. I think that could be one of the reasons I think we see this problem. That is my observation.
Okay.
Vijay sir?
Well, I think 2-3 points quickly I would like to mention. First is that there is a difference between the VAC, that is, the ventilated air conditioning system, or even heat induced system, and the AHU, which is slightly different, which is made for the clean room. So we have to consult, as very rightly said by our engineer, that we have to involve a biomedical engineer and explain to him our purpose and the goal behind what we want to establish. That will make a lot of difference, because regulating the air output is a must, along with the air quality check. So depending upon your native count, first you analyze totally what is the native count, and then you have to decide what kinds of filters, how many filters you'll require, whether along with the HEPA filters, you also need 2 or 3 pre-filters, Also, you need to incorporate any of the potassium permanganate plus activated. These are all the technical things which I think an engineer will be able to tell you in a better manner after analyzing the native air quality. That is a must. Then outcome, as you asked in this particular question, outcome in what sense it is not optimum. If it is a pregnancy rate, then there can be a lot of aspects which you look for. But if it is related to the air quality, then again, you have to first look for the native air count. Then what are the things that are generating the air pollutants? What kinds of pollutants are generated? Then inability of the filtering system. Why the filtering system is not incapable of taking care of that one? That you have to study. And then non-maintenance ducts. Duct maintenance is one of the factors which is totally ignored many times, because either it is somewhere going through the ceilings or outside, which is non-accessible. These are some of the technical aspects which we have to consider.
Yeah.
That's a good point, Dr. Mangoli, because just having an AHU system will not help. Making sure that it's functionally working and
accessible,
the required quality of air, is because most AHUs without proper maintenance will break down and will prove to be counterproductive.
Absolutely. Absolutely. And we'll touch upon this in the later slides.
Can I come in a couple of points on this?
Yes.
First of all, thanks for involving me in this discussion. Thanks to iHera, thanks to Life in Vitro. And I was thinking like being only engineer in this group, but from the discussion, what I realized is there is hardly any gap in the knowledge, So many good comments are coming in, and probably most of the points are covered. I was part of this discussion and was listening about the points related to clean room, and I just wanted to share here that again, clean room is not something unique, or clean rooms are again various type of clean rooms. You can talk about clean room in a pharmaceutical manufacturing facility. You can talk about a semiconductor clean room also. And though both are clean rooms, they will have different specifications. So here, as other panelists rightly mentioned, while we are designing a clean room for IVF laboratory, we will call it as a clean room. So the different inputs required for IVF point of view has to be incorporated. So that is the only thing when we do, I think we can get the right solution. And in the previous slide, the question we were talking about, I think something similar was happening to the word air handling unit. So again, AHU is a machine which is giving us desired air quality, but that has to be designed according to your requirement. You have air handling units installed in automobile facilities, you have air handling units installed in your malls or offices, as well as in IVF lab, and each AHU will be different in terms of its filtration components. So I think most of these points were already covered.
Yeah.
So so we have to consider our specific. One is location specific pollutants. That will be dependent on the location where you are going to have your laboratory. As we mentioned earlier, that somebody is probably installing a facility today out of a metro city, but tomorrow it becomes the air quality changes. So those are the aspects. And second aspects will be what you want inside. What is the requirement indoors? So accordingly, your AHU has to be designed, and then we can get better results.
Okay, so Upendra, let me ask you one simple question. So I'm a prospective person who wants to open a lab. And so as per what you said, location specific, I need to first find out that what are the level of pollutants at a particular location where I'm going to build a lab. So that would be a good way to start, to know what are the ozone levels, the SO3 levels, and then you mean to say that we can actually design the lab in a way that we can address these particular points and try to bring them down?
Yes, I totally agree with the point. So I would not say design the lab. I would say design your air handling system.
Correct.
So yeah.
Yes.
If we are aware in the beginning, we can avoid retrofit related troubles later. So it's the best way.
So if we know that the area is has a very high concentration of VOCs, then appropriately we can make the AHU, robust enough to
accordingly. Exactly.
Great. Okay. So Keshav,
that's a fantastic point which Upendra has put, and thank you, Gaurav, for actually bringing this up. And obviously, yes, if you're designing an AHU for like all these different kinds of facilities, like a microprocessor unit, an AHU which takes up to like 40 exchanges, but in IVF laboratory, a 40 exchange unit would not be like of any use. You probably are happy with about 15 exchanges, etc. And also you'll want to look at the pressure, etc. So there's so many things that need to be checked. And again, the VOCs that are there in the area and in the environment, you can actually have specifically designed filtration systems for the specific location. Yeah, let's move on to the next one.
I just, before that, I just want to make a comment over here. Okay. In terms of your environmental component, it is not static. Please see, while we say that, okay, we can design AHU for handling environmental VOC component, please understand that your VOC components are not static. It is ever rolling. So today, if there is a heavy rain in some place, the static points will change.
Yes.
So we need to have, you need a dynamic filtration system, not a static filtration element. That requires a lot of discussion. I don't think that we can complete over in this session, but we can have a detailed study on this.
What I can take home from the information that I've gotten here is what Dr. Sangeeta said, that in a particular city where she feels that the environment is different and is posing a heavier load on her filtration systems, she's going ahead and changing the HEPA filter times a year, which is unheard of. So she's customizing her air handling unit to the demands of the location where she's located.
Yeah. So, but you can similar way you can ask Upendra Sethe. I think Upendra Sethe also may able to comment on this. Upendra Sethe, you can ask on him. The HEPA filter in some of the other clean rooms are changed every 90 days.
Okay.
So it depends on what industry you are in. And at the same time, you may have in the backup, you may have a molecular filter, which is totally different for using for something else. Upendra may comment on this.
Yeah. Dr. Maju, thanks for bringing a very important point. It's very absolutely correct to say that ambient air quality is not constant. It changes as per season. It changes along with your neighbors. Sometimes some industry may come up, some facility may come up and air quality changes. And yes, your seasonal changes. If we talk about northern part of India, winter season, we find a very different air quality from particulate matter point of view. If we talk about western part of India or southern, with monsoon, a lot of polluting gases will get cleaned up automatically. So, most of the times when we are designing air conditioning system, we will try to work around known worst case scenario. And in parallel to that, continuous monitoring of the system, that helps us, adjusting these fluctuations in the air quality. So, as Dr. Sangeeta mentioned, that she has to change HEPA filters every three times a year. So there, some modifications can be done. You can look at primary filters, because HEPA filter is your last line of defense. And before that, what is it protecting HEPA filter? As Dr. Jacob mentioned, that the molecular filter before it, primary filter before it. So we can design, some filters in the system which can take bulk of the pollution, and they can help you cushion life for the HEPA. So, but despite of all this, regular monitoring of air quality, you cannot run away from that. So all this.
And these are cities where pollution levels are nowhere close to what we face in India. Yeah. So that's also something that we should note. I think we've already covered this question amongst the discussion, so we should move on to the next one.
So as for this question now, we want to know whether a single AHU can suffice for both the IVF laboratory as well as the adjacent IVF OT where egg retrieval is done. So what would you suggest? Alex, you could take this.
Yeah.
Should we have a single common AHU or separate AHUs, and why?
See, there were some discussion about it in some of the interactive meetings. If the engineering team can design an AHU which can cater to the needs of both sides, it is okay to go with the same AHU. But with the kind of expertise that we get in our country here, I think it is ideal. I think in many countries, I think it must be the same thing. It is always better to go with a separate AHU and on dedicated AHU for the IVF laboratory, so that you won't be mixing the pollutants or the disinfectant coming from the OT to the, because nowadays you see that if you don't have proper pressurizing system, high pressure, even you can get the high VOC when the anesthetic gas is being used in the egg retrieval time. So I think ideally it should go with a separate AHUs for.
So that's a very important point, that the anesthetic gases are a big source of VOCs, which we tend to overlook and we tend to forget about. Because you could argue that no disinfectants are used, or all disinfectants which are used in the OT are also embryo safe. But what to do about the anesthetic gases?
And second important thing is that you can see, I think 95% of the clinics in India still uses the box incubator with 5% or 6% CO2.
Yeah.
And rest 95% of the air inside that box incubator is derived from the room air. So whatever is in there, that is reflected in your, that finally dissolve into your oil, finally into your culture system. And if your culture system is a micro mode of culture system, the more concentration will happen to the micro droplet.
So then you are saying that it is a very good idea to have a separate AHU for the culture room.
Yeah. Can I add something?
I want to bring in Gaurav here because I want him to play the devil's advocate at this point, because like he has experience in working at centers without AHUs and still giving fantastic results. So how do you do that?
Yeah, that is my question to the panelists here. You have a control arm here for your RCT. Where if I can argue with Alex and Dr. Mangoli here, who are working in centers with dedicated AHUs and clean room environment, and they're constantly monitoring their VOC levels, and at the end, I would think that these are good practice, points. But when you have to measure success between a laboratory which has these provisions and laboratories and majority laboratories which don't have these provisions, but if the KPIs are being achieved, if I can say that I have a very good percentage of expanded blastocysts and my implantation rates, so if my pregnancy loss rate is equivalent to a laboratory in Norway or in Scandinavia, where the outdoor air quality is perhaps better than the air inside our laboratories, then that's a very strong argument.
Okay, Gaurav, here I would like to come in and then comment on it. See, around 15 to 20 years ago, we also used to work without any AHU. And we used to get good pregnancy rates depending upon whatever at that time persistent. What is more important is the consistency in getting these results, over a long period of time. That is where the AHUs, come in contact. AHUs, one thing you remember, that installing AHU will never have any negative impact unless and until there is something wrong in the system itself, and the AHU itself is pouring in, wrong type of air into this one. So these are the extra precautionary things that will help you in getting consistent results. Now, as far as this part is concerned, that is whether you should have a single AHU or double AHU, I would like to comment on this, that it depends on two factors. One is the area of the culture room and the OT and the capability of that AHU to handle the air rate per hours. So it depends upon what is the pump's capacity. And second thing is whether both culture room and OT are connected through only a small window through which one passes the tubes or the petri dishes. Because you remember that if everything else is made up of wall and there is only a small window available which intermittently opens, there is absolutely no use of having a single AHU fitted for your culture room, because that will never have any communication with the OT. On the contrary, if the OT and culture room are adjacent to each other with a huge door in between where the air exchange passes, then there is a possibility that you can create enough positive pressure in the culture room and the air can go through from culture room to OT to outside. So these are the two factors I think important. Maybe some engineers will throw some light on it.
So do we believe that having a positive pressure unit with, let's say, a normal HEPA filter can substitute an AHU?
No, not at all.
No.
So that is why AHU also have, for example, if your complete center has a purification system, but there are different definitions of purifications for different reasons. That is how it should be engineered.
But sir, install like an AHU in a small laboratory, which is like, let's say 10 by 10, which is very common size for laboratories, is something which doesn't make a lot of sense to a lot of people. And then they'll say that, okay, we can buy a small, let's say a Cora tower or a air filtration unit, and that should suffice. Correct. How do you counter that?
Correct. It exactly depends upon the capacity of your AHU unit, how efficient they are and how much total area it covers. What you say, Dr. Majum, Jacob?
Yeah, see, thing is that I don't know whether I am, if I tell something, it become very sarcastic, but one thing I want to tell you is that nobody can mix up air between two rooms in any clean room concepts, because otherwise both the rooms should be of same standard. That's the simple thing. If I'm having a class 10,000 room, then the next room is class 10,000, then I can both the rooms. But the challenge on the engineer is that how will I balance these two rooms? I have to balance the airflow between the laminar flow. If you see a laminar flow which is happening from the all four corners, like the two different chambers to balance the airflow in all the corners. It requires a lot of high technique ISRO type of labs where we have some other fluid dynamic system used to balance between the rooms. That's too expensive than buying two different AHUs. And second thing, I want to, in this group, I want to tell that AHU and split AC, the difference of pricing is very, very thin line. The thing is, what cost you is the filters. It's not the AHU. AHU is not a very big costly item. In this, what cost you is the plenum, what cost you is the filter. A good plenum cost you more, a good filter set cost you more. So thing is that as an engineer, it is a challenge. As a technologist, it's a challenge to balance air flows between two consecutive rooms. And to balance the air, we need four walls. We cannot have a partition in between that wall. So that's a technical challenge, unfortunately. So if you want to mix up the air, that's all clinical issue. I don't think that mixing up air between different quality air from another room to different quality air to other room will not give your required air quality in both the rooms. So that's what technically I want to tell you.
Okay.
Right. So,
Dr. Keshav, maybe one small point. As Dr. Jacob very correctly mentioned, that AHUs, it's not always true that AHUs are very expensive. So it depends on what capacity and what all you want to be installed in the AHU. And when we are trying to handle two different rooms with two different cleanliness requirements, what will happen is we will have to design AHU for the cleanest room, for the room which allows minimum level of particulates and gases. So in a longer run, when you are looking at capital plus operating cost, probably you are using more expensive filters and replacing them often. So I think separation is generally a good idea. It depends on the site. If the areas are too small, then it's something else.
Mr. Jacob and Upendra, here the objective is to make sure that the outside impure air does not enter the IVF laboratory, the culture space, right? We are not about the mixing so much.
I understood.
Just making sure that the external influences, they don't come inside the laboratory.
Exactly. Yeah. So I understood, Dr. Gaurav. I want to just give you a quick answer to this. See, to set different pressure levels in different rooms, I need different AHUs, or I need to have different air control units. Okay. So the ideal thing is, what I would say is, the highest pressure level should be in your ICSI lab or IVF lab. The next level should be in your ICSI OT or whatever the aspiration place. And the lowest level should be in your corridors.
Different AHUs or a single AHU?
I think it can be done with a single AHU. I think it can be, Jacob, I think it can be done with a single AHU if you're maintaining like those air exchanges in the laboratory itself. That'll create a positive pressure in the laboratory. And if the outside area does not have it,
no,
absolutely yes.
I'll explain you that technical difficulty. See, whatever the air which you are pouring into a room and the other room, both is going to come into the air handling unit into the return. The return is going to go into the same place. So you see an air handling unit. Then what we do is we reduce the returns from different units. So different rooms returns we reduce using a volume controller, VCD, volume control device. So that volume control device is very, very flimsy thing, which normally go wrong. However, today also it is the 7th time I am balancing my lab today. So to get the exact balance. So it's difficult to use that air VCD device to actually balance the air and control a pressure difference between two rooms. But if it is two different HU, what I can do is I can control the speed of the fan, speed of the blower, and adjust the pressure. That becomes more effective way of managing it. It's just simple principle of fluid dynamics. But yes, you are correct. I can do balancing, but it is tough task. Task is much task. And it can go, sometimes it can go wrong without my knowledge.
Point taken.
Point taken.
Yeah.
Great.
And
so
second thing, second
Alex.
thing, like what we see, and if it is a hospital-based IV unit or anywhere, the maintenance engineer, maintenance people, how often you can get them if something is going wrong. So suppose if you have a dedicated on IHU for your IVF lab, you just forget about the things. So you are at the peace of mind. So that is one of the things. So otherwise, if anything goes wrong, I think it has to be, the total thing need to be rearranged or need to be repaired.
So that's the important take-home message for the audience. 2 separate AHUs, one for the ICSI lab and one for the OT, which could also be a common AHU for the hospital.
Yeah.
And this will help you in making sure that the pressure in the embryology lab is the highest, so that air, if at all, if any air has to exit, it'll exit the embryology lab and not enter. And then the next pressure is in the OT, and then the lowest pressure is outside. Okay. So let's now, we'll take this question about positive pressure and how do you retrofit existing lab with positive pressure, because we were unable to do so in our laboratory. And a lot of people have this problem across the country, where they find it almost impossible due to location constraints to retrofit their labs with a positive pressure unit.
This is one of the things that we mentioned in the Cairo consensus in the document. They had mentioned about it because it is not possible in some of the laboratories, so they have to use the standalone AHUs. So that, but only thing is that they won't be able to get the positive pressure. Okay. I was discussing with the Sathe because in India, most of the clinics, I think more than 50% of the clinics are batch IVF centers, maybe having a 10 into 10 feet or 10 into 15 feet IVF laboratory. So I was asking you, what is the cost-effective method? You can have a positive pressure, you can have a cost-effective AHU, and also chiller or maybe AC unit. Maybe Satya can discuss something about that.
Yes, sir. Thanks. I think this is a very important point. When we are talking about any clean area, positive pressure is very important. And while we are trying to maintain it under positive pressure, we do not want to disturb other parameters, which are temperature, humidity, and pollutant level. So for positive pressure, typically the standalone units will suck the air from room, clean or cool it and throw back. So they are not able to provide that positive pressure. So for maintaining a positive pressure, we need to continuously add some quantity of air. This quantity depends on your room size and how much positive pressure we want to maintain, how much properly sealed it is, and that has to be cooled and filtered first. So a cost-effective way, as Dr. Alex mentioned that we were discussing the other day, is cooling becomes a challenge, and it is considered that you need to have a massive chiller on the roof of the hospital, and then it becomes a very expensive thing. But this can be optimized based on the room size and the small amount of air which you have to bring in. That can be cooled with our outdoor units. Typically, we have split ACs installed in our air conditioning places, maybe office or clinics, and you have one unit inside and one unit outside. That is called as DX unit in air conditioning language. So those kind of compact fresh air units are possible to be installed. They are not too expensive, and that gives you a freedom of if you want, you can shut off for some time also. So there are solutions available, and that can be implemented.
But in an existing laboratory which does not have an AHU, which does not have those ducts which are there,
correct,
it becomes such a big challenge.
Yeah. I think where you do not have ducts, the distribution of air becomes a challenge because, if I am bringing in outside air and just releasing it in one opening, make one opening in the wall and releasing in the room, there is a problem that, I am creating a pocket of, call it clean air, maybe it has different parameters. So that has to be distributed uniformly. So then the ways are, you have a false ceiling and a small duct running with 2 or 3 openings. Alternate ways can be, if you have very small areas, probably you can have a couple of wall openings and duct running outside. So those will be site-specific solutions, but they are possible. These things
or you basically convert it into a modular laboratory. Obviously, it'll take some time, but that's something where with the false ceilings and the panels, etc., all that can be done. Okay, let's move on because we're actually running quite ahead of time. There's a lot of discussion on VOCs and the effects of VOCs on embryos, and I think all of us know about it. So let's try and discuss a little bit about how to manage VOCs. So the first question here would be, how do you actually measure VOCs in your laboratory? How frequently do you do it? And is it actually necessary? Alexa, you can take this question, and then we'll move on to Vijay sir. So you have to mute yourself, unmute yourself.
Yeah, sorry. So as per the Cairo consensus, the VOC level in the lab, the threshold level that they mentioned is the cutoff value is around 400 to 800 ppb. And if you see most of the labs that we see in India, I think they are using a ppb-based photoionizers. So I think lowest detection level is maximum is 1 ppm. So 1 ppm is equivalent to 1000 ppb. So I think we can see in many of the lab, if you visit, they give us some kind of a backlight, like a green, orange, or something like that. So I think that has to be taken care of. If maybe some of the Indian companies, they are using a kind of a quality control package where they bring in the PPB-based meters, so people can use that kind of system. Yeah.
And sir, Vijay sir, I'll get you in here. How frequently are you actually monitoring VOCs in your laboratory?
Well, actually measuring the VOCs, as Alex said, that, these are the criteria. There are criteria in different units in which VOCs can be measured depending upon the sensitivities. So ppm and ppb is one part of it, but if you see, actually VOCs are not part of the particles that can be measured, but it is in the vapor phase. So actually, sensitivity-wise, many pharmacological industries, they measure it in mg per meter cube. And they have a criteria of the TVOC levels, that is less than 0.3 mg per meter cube is low. Between 0.3 and 0.5 is acceptable. Between 0.5 and 1 is marginal. And 1 to 3 is considered as very high. But yes, we have given a second thing is measuring VOC is very critical, and there are VOC meters available right from 5,000 rupees to 3 lakhs rupees. So it entirely depends upon how sensitive the sensors are which are built in there and how reproducible results they give. So basically, we also previously had this 20,000-30,000 rupees VOC meters which are available, but I'm not very happy with that, because giving it a green color is a very crude way of of evaluating your air quality. That will not make much difference into your air monitoring systems. So now we give it with outsource, and every 1 month or every 2 months, depending upon your workload, we ask the company to come and then measure it.
Okay, I just want a quick clarification because it came up on the chat. Alex, can you just repeat that a VOC meter, so we need to buy a VOC meter which measures in ppm or ppb?
I don't want to recommend that thing, because it's a very costly affair. If you go with the ppb based meters,
okay,
it can cost up to maybe 4 to 5 lakhs.
Okay.
Nowadays, you have the QC companies. I think that part of QC, they come in and measure all your QC parameters. Along with that, they also measure the ppb level. I think that would be okay for many of the laboratories, and maybe one can keep a ppm-based continuous monitoring system so that they can get an idea.
So ppm is less sensitive than ppb.
No, ppb is more sensitive. ppb.
Yeah. So ppm is less sensitive.
Less sensitive, but you can get an idea what is the level of the VOC in your laboratory, so that continuously you can monitor.
All right.
And yeah.
Thank you.
And maybe Satya can give a more insight how you can analyze with the GC, gas chromatography, and with the 10x, and get the air inside a cylinder, and maybe once in a year.
Yeah, Dr. Alex, thanks. So I think I totally agree with the points you mentioned. The handheld devices, and there is a range of devices, and as the sensitivity is high, they are expensive. Also, I think they have some sensors which has to be maintained later on, so those kind of costs are there. So as it was discussed, I think Dr. Mangoli also mentioned this, that there are certain laboratories who do gas chromatograph GC analysis. They use MSGC, which is mass spectrum gas chromatograph, and they send the probes to your site. That probes are basically tubes with reagent and a small pump. These are very compact devices, and the results are very accurate. So those devices can be in your site for few hours. And few recommendations are, it depends on if you are operating at different shifts and workload is changing. Probably they would recommend 2 or 3 sets of reading, but those are very accurate readings, and that can be done at interval of say once in 3 months or something like that. And a simpler handheld device can help you keep an eye that something is not going drastically wrong. Suddenly you are shooting into ppm and you are not aware. So I think that combination will be good. And these GC-based lab reports are very accurate, and they can help you designing your systems and monitoring continuously. So combination is a good idea.
Okay, excellent. Now, quickly, can the panelists tell us the important ways how we can bring the VOCs down in the ambient air? So one by one, just very short answers about what do you, as consultants, what will you advise? What are the things to be used in the lab to bring down your VOC level?
So if you have a dedicated AHU system, then definitely you can integrate the different type of VOC filters, starting with the activated carbon or activated potassium permanganate, and even activated alumina, because the activated carbon doesn't alone absorb all the the acid and other, the aldehyde and everything. So I think you should have a combination filter for VOCs, and also maybe there are different systems like maybe photocatalytic oxidation systems also there now who can retrofit into your ducting induct systems are there. So I think these are some of the area you can, and along with that, the safe practice.
So this you recommend in the duct?
Yeah, in the duct. And also if you don't have any HU, maybe you can have your standalone towers with all these maybe systems. Yeah, that is only way.
Okay, so a combination of, so just carbon filters will not suffice?
No.
What is the other two filters you mentioned?
The one is potassium permanganate, another is alumina, activated alumina.
Activated alumina. Okay, so that's something to look out.
I'm telling few. I'm telling few. Maybe I think Mr. Ubendra, being a chemical engineer special, so he can put more light on that.
And you and Dr. Mangoli, you also use something for the gas lines for the.
Yes. Yes, inline filters, we use it. And plus we have also had special filters fitted like activated carbon plus potassium permanganate, you know, as a part of the HU itself. But what I would like to say is that it is better to avoid the factors that are generating the VOCs into your laboratory. You have to keep a constant eye on those factors. For example, if someone coming with the perfumes on, if someone has put the mehndi on their hands, if someone is keeping, if you have kept a battery for your UPS systems nearby, these are the factors which creates a lot of VOCs and negative elements and then pouring into your air quality. So we have to take care of such things that will eliminate or basically minimize, so that it will be less taxing on your whole air purification system as such.
All right, that's a great point. Okay, one quick question here. I have a laboratory now. Hypothetically, I have a laboratory where I culture in triple gas. So there is no exchange with the ambient air. And I have very nice laminar flow hoods where hoods with not just filtration for particulate matter, but also with carbon filters. So to make sure that while manipulation is going on in vitro, they are in a safe place. Do I still need to worry about the VOCs in the ambient air?
See, here what you're talking about, the tri-gas environment, like the mixture of oxygen, nitrogen, and the carbon dioxide. But you have to keep in mind that even if you use inline filter, the gas that you get in India, it is not a really pure gases. And I still remember I was helping one of the clinic to set up the design, what we were discussing. And they had all the carbon filter and everything in the unit and a nice ISO 7 type of a thing. And initially for some time when we are culturing the embryos in the benchtop incubator, that has got a regulator for nitrogen and oxygen. So the pregnancy was not happening properly. So after that, the culture started in the box incubator with only CO2. Then they started getting the pregnancy. I'm talking about a batching center.
Okay.
So you know what really here happened. It is not only your ambient air condition, it is also the type of gas you are getting. That is a real culprit in many cities.
Okay, that's a good point, that the quality of gas is as important. But I would again like to ask you specifically that if I make sure that there is no gas exchange between the ambient air and the incubator culture space, then should I worry about the VOCs in the ambient air?
Because you have to open it, because it depends on how many times you're opening your incubators. Okay. Even if you're using a benchtop incubator, it is open top loading incubators. So
Dr.
even there was a study, yeah.
Dr. Vijay, what do you think? Should I still be highly worried, or is it still all right if I know that there is no, all my entire culture is in benchtop incubators with no exchange?
Right, but still I think it is difficult or not possible to avoid any kind of exchange between your ambient air and the your culture media at some point of time, whether at the time of equilibration, whether at the time of handling the oocytes, gametes, embryos, you are going to expose it. So worrying about the VOC is important, because even if it enters into the smaller amount, as you are handling the smaller droplets, it proportionately it will make a large difference. As far as the gas and the composition and the purity of the gas is concerned, are we going to cover that point next? Otherwise, I will come.
We're going to come to it.
Oh, okay, fine. So I'll comment on that line.
And it's been rightly mentioned in the chat as well. The chat also has been very active. So thank you to all the participants who are constantly engaging in the discussion. So at the time of like media preparation or dish preparation or even like opening the media bottles, etc., you will have like the VOC being mixed with these media.
The VOC will come into play at that time.
Yeah. Cool. Yeah. Let's talk a little bit about the particulate matter we've spoken. So let's have a small discussion about the particulate matter. We are running really ahead of time, so please be very brief. What are the particulate matters that we should be worried about, and how to actually overcome like the challenge from them?
Yeah, there
Vijay,
are some. Please
Alexa, you take it. Yeah, Alexa, you can take it.
Okay, fine. Because initially, before these activated carbon, the studies were done. Even I think Keshu was shown on slide on earlier study showing the effect of the PM 2.5. I think this year there is a study came out from one group that was published in a toxicology journal, reputed toxicology, I think. They looked at the mice model and they infused in the trachea the PM 2.5, and they found the oogenesis and in the blastulation rate and everything is getting affected. So I think the particles are also very important, especially I think the PM 2.5 and those which is less than that is also very important to be taken into consideration.
So Vijay sir, are you happy with a HEPA filter, or you'll think of using an ALPA filter? I've never used an ALPA filter. We've only done HEPA. So.
Yeah, I think HEPA filter in most of the cases it will be enough, but it again depends upon your environmental, pollution level and the dust particles that are floating around you. So, as far as the normal levels of the 0.1 or even 2.5 parts are concerned, you have to slightly take a little bit more precautions from using the HEPA filters. You can do that. But what I want to insist is that the sources of again I want to talk about the source part of it, because I have seen in many laboratories that the staff, the embryologists who are working in the culture room, they go with the same clothes on to the pantries, to the libraries, to different places, and then come and start working on their workstations. I think this gets a lot of particulate matters along with the bacterial contamination problems and all these things to your culture room. So it should be made compulsory and mandatory that you should wear a gown, sterile gown, before entering into your culture room, so that you are going to take care of it. Plus the papers, plus all the things that may carry the particulates without unknowingly, that has to be taken care of.
Yeah, I think that's a very valid point. Gaurav, we've actually covered the first 2 points of this question. Maybe we can take this third point and move on.
Yes. So isn't it enough to have a portable air handling unit in the IVF lab? So what are your views? We've discussed this now.
Yeah.
That, it's preferable to have an AHU, but if you don't have one and it may be difficult to retrofit labs with AHUs, then you have to rely on these. A very interesting thread is going on in the chat box, which I was just reading. So I'd like to pose that quick question, where people are talking about putting open charcoal in the laboratory. So what are your views on that? So there are very interesting responses where people are saying that actually might be detrimental, or it is ineffective, or what is your take on that?
Well, you obviously cannot keep an open carbon particles into your, to absorb the, or to pass the air through that one, because ultimately that will really become more detrimental, because unknowingly, because of the air ventilations, a lot of air changes, that is also going to take part of this particles from the charcoals and then going to spread on it. So if you have to keep it, then you have to cover it with a kind of filtration system on it.
Oh, and okay, that's very important.
Here I want to make a point.
Yeah.
I want to make a point. See, when you are using a charcoal filter in any of the pre-filter stage, it's important to use a semi-HEPA after the charcoal filter. because charcoal, in whatever methodology you pack it as a filter, the emission out of the charcoal of microns particles or charcoal micron particles ppm will happen. So please make sure that there is a semi HEPA immediately after the charcoal filter.
Then open charcoal is a bad idea. Open charcoal in petri dishes is a bad idea. Am I correct?
Yes.
Yeah. Even I have a small point here quickly. Though activated carbon, though carbon is an agent which is going to absorb it, but you have to manufacture it properly. The filters, what are there, they have bonded, they have manufactured into granules. It has to be activated so that steam activation is a process, so that all the pores of the carbon are available for the adsorption process. As Dr. Alex mentioned in earlier answer, sometimes they have impregnations based on what pollutants you have to remove. So, you get overall control on the process. So that's why a tested carbon filter is a good idea, so that you know what you are installing. And as Dr. Jacob mentioned, as Mr. Jacob mentioned, after any carbon filter, HEPA filter in the end is a must. Because yeah.
I might be very wrong here, but like from my understanding, if you're putting charcoal in the open petri dishes, I don't think it's going to help with the VOCs. The only thing that it helps with is humidity. Am I right, or am I wrong here? Because the air is not circulating, it's not passing through the filter. Exactly. So it's only going to help with like humidity control.
Correct, Keshav. It will absorb it.
Yes, humidity will be absorbed, but as you rightly said, air is not going to air is not circulated over there, so it may not be cleaning entire room and only a small pocket. Probably it is getting humidified, and that process is not, you do not have control on the process. So
yeah,
actually, what
absorb some orders if you are having it.
But this is an important myth which has been dispelled here, that because I have seen this so many times, people are actually putting charcoal in open dishes in their incubators. This is not that's not going to help, because what I understood is that it's not activated charcoal, and B, it has to be sequestered, because it may be counterproductive, because it is releasing stuff which is highly detrimental.
And also maybe it can absorb maybe CO2, so that your CO2 concentration will go down. The box incubator.
Even
maybe
more.
Maybe they can, what they can do is that they can put a pre-filter in the LF hood, laminar flow hood. Yeah. Nowadays you get carbon filters that can be kept as a pre-filter in your IVF workstations.
Yes,
absolutely
using one of those.
Yes, in the inlet, in the inlet side, not on the mid segment. It should, the inlet side, you can have a carbon impregnated filter. Impregnated filter, not a carbon direct filter.
Yeah,
should be on another fiber. It should be on another fiber. Second is, please understand that carbon or the activated carbon is absorbing water particles, and along with the water particles, other things are being absorbed. It is carbon absorbs everything. So carbon is not particular for a particular VOC element. So if you are looking at a very particular VOC element, like what Alexa said, maybe alumina or potassium permanganate has to be looked in, where you may have some particular VOC elements has to be absorbed, which doesn't get absorbed along with the humidity control. So the carbon filter do two purposes. One is the humidity control. Second is the VOC, other particles absorbed. These are the two purposes of the carbon filter.
Okay, perfect. So let's move on. Keshav, I think we've talked about this, the AHU being a double-edged sword. So
yeah.
The next one.
You want to talk a little bit about the gas scenarios in the country? I wanted Vijay sir to mention it a bit, and he wanted to do it.
Yeah. Yes. Can I go ahead?
Yes.
Yeah. Okay, I think medical gas manufacturing, this is extremely serious issue, and this has to be regulated at the government level with the equal precautionary measures as applied to the drug industries. Because see, the basic requirement for the gas production units, they have to control. They have to control the qualifications of the personnel who are manufacturing the medical gases. If you see those units where the people are manufacturing, you will be aghast at. Written commitment, there has to be their liabilities, their written commitments, their quality control testing protocols, their certifications, their validations, regular counter checking by the government authorities, whether whatever they are claiming they are producing, whether it is a mixture of the gases, whether they are purity of the gases, it has to be a controlled industry. I strongly believe in that, because IVF labs should maintain their own cylinders, including spare parts. You cannot exchange it with the cylinders. For example, you have to keep your spare cylinders also with you. You purchase them and keep it with them. Generally, what people do is, once my cylinder is empty, we hand over it to him, and he may supply it to any industry. You don't have any control on that. But this has to be written. The name of your clinic has to be written on each and every cylinder, and you should insist that my cylinder will come to me only. That is a very must. I think that precautions has to be taken. So it should be never.
So what about having like common gas lines within the hospital which supplies CO2 and nitrogen, and having like a, so these supplies are there for the OT as well. Now that can be very, very detrimental, because we don't know which cylinder is being put, etc., etc. So
I will always prefer that IVF should have a separate line.
But when a hospital goes in for NABH, the NABH certifiers come and say that, why are you using separate cylinders? Why are they not being provided by?
Yes, you should be able to explain them, Keshav, properly.
Yeah.
No? This is what the explanation that you can give. This may be quite detrimental, and I strongly believe that we should have a separate. We should have a control on the quality control.
Yeah. Okay.
Okay, Keshav, you take this one.
Okay, so this actually this image gave me nightmares, because I have not looked at the AHU vents for quite a long time now. I'll be very honest with all of you. And when I look at this image, I'm very scared to actually now go back and open up these vents, because like if an AHU is not maintained properly, if the ducting is not maintained properly, they are like prime sites for fungal growth. And that's something that quite a lot of people overlook. So your comments on this, and then we talk a little bit about microbial contamination in the laboratory and end this session.
I think since we think that HEPA filters can last long, because we bring in the third party and just check the integrity of the filters. But the problem is that, especially since we live in the tropical countries, these HEPA filters can become the breeding ground for the mold, especially Aspergillus or any bacteria. That is the same case with our laminar flow hood HEPA filters too. So that's why even not only I think today we heard about from Dr. Jindal, she's changing 2-3 times. Even from Middle East, I think on group, a big group, they are also telling that they are changing the HEPA filter twice in a year. I think that thing has to be taken into consideration. And especially there is something known as a sick building syndrome. In some old building, when you enter with the HVAC and everything, you get the smell. Basically, that is because of this mold growth and the VOC generated from this thing. So this is need to be taken into serious consideration.
Yeah. And even in batch scenarios, like, okay, you have a setup, you have a laboratory which has an AHU, but once the batch gets over, you switch off the AHU.
Yeah.
In those scenarios also, these are prime sites where you will get fungal contamination. So I think a message should go that the AHU should be running continuously.
Yeah. Maybe also, maybe they can put the UVC, if UVC lights. I think that is maybe with the pandemic, I think that will be a good add-on. I think,
And on the contrary, I'd just give an anecdote where for a particular laboratory, the moment we closed down the AHU, all the things fell back into place. So,
yeah, so that's a very good scenario where the AHU is actually introducing problems into the laboratory. So
that is what
is the most counterproductive thing in that IVF laboratory.
So it is a double-edged sword, I think. That is what mentioned in one of the slide. So if it is not done properly,
I think maintain the shortest distance between your input in light of the incoming air and the actually duct. The duct should be as short as possible, and plus, as Varghese very rightly said, that introduce the UV part of it, make it of complete stainless steel washable, and make it of different parts. What we did is, we faced the same problem previously, so we had divided that, and now we can assemble it, so that we can take out in between parts and then clean it and then do it. So the cleaning became very easier right now when we separated the part of it.
Okay, those
Yeah,
are
see,
very
exactly. Dr. Vijay has put the nail on the right place, and I'm saying that. See, one is that in the descent stage, which you need to think about is that the duct length, especially your supply and return duct length, should be very, very short, as short as possible. Second, you should have an access to your ceiling, not from the lab, from outside, if possible. If possible. Otherwise, all the ducts should be able to disbandable and pull out. In whenever required, it can be pulled out and pushed back.
from
This
outside,
will help you
from the top.
If it is from the terrace, it is possible. I am talking about from the terrace, it's all possible. If the entire unit can be lifted up. But I'm saying even on the in-between floors, like floors in the second floor, third floor, where there is another 5 floors above. Even there you have a possibility of designing pull out ducts and pull in ducts.
Okay.
And the plenum pull in and plenum pull out and shortest distance. Even see, everywhere in India industry, you will see that you will have only square ducts. Why can't we have cylindrical ducts, which is having less dead space to grow any microbacterial things? So it's important that one is that you should be able to do a reverse flow of your circulation. That also, because when you do the cleaning process, I prefer to have the air flowing from the return to the supply, so that everything is reversed, and then I am going to go the cleaning process like that. So once in a year, if you are shutting down your lab for 15 days or 12 days, you should have a reverse process of cleaning it up. So shorter duct, reverse process of airflow, and removable parts of ducts in the design is an essential part if you want to have your ducts cleaned properly.
Mr. Jacob.
Absolutely. Yeah, I agree with him.
So, and what are your views of having the AHU on the terrace, vis-a-vis having it somewhere in the building?
Oh, see, I would like to have the AHU to be placed in a more circulated place than a closed room. So it's better to have on the terrace, because you get more ventilated. You can place, because my fresh air concentration of VOCs will be lower when I go up. And second, if it is fully ventilated.
Okay, excellent.
The VOC concentration inside the area will be less.
Yeah, but that's where the challenge of like forest fires and all of those contaminants comes into play, because if something like that happens nearby, then it might be introducing more pollutants. Obviously, it's going to be a rare scenario.
You need to shut down your fresh air. You need to, if you seeing that, immediately you need to shut down your fresh air. And since your place is fully ventilated, in the time which is going to go out of that particular area, it will go out. Okay. But think that if it is in a closed room, it enters the room. To evacuate that, you have to do another process. Maybe you have to bring 3 or 5 suctions, suck everything out. All this process you have to do. But if it is in an open air, it will blows out. Only thing, you have to go and keep your fresh air shut, and maybe the AHU shut, and put on the standby. Standby split or whatever.
Excellent suggestions. Thank you, Mr. Jacob.
Yeah. So moving on to the last part, I think Gaurav, you want to take the microbial monitoring, and then we move on to the recommendations from the panel.
Yeah. So Alex, we start with you.
Yeah.
That what are your suggestions, recommendations for microbial monitoring in IVF laboratory?
Yes, see, what we do in a craft, what we do is that before starting every month, we do the settle plates and the active settle plates. So we have that high media, the aspirator. So aspirator, high media, that a small machine, you can aspirate the air from the room, and you can keep that dish inside that thing. I don't remember
How
exactly.
often do you do it? How often do you do
it? Every month we do that.
Every month?
Yeah. So since we have the in-house microbiology laboratory, so we do that. So that way we can make sure that we are on a safe side. Yeah.
Anyone else wanting to comment on this?
The main thing the microbial contamination that may come into is because of the storage part of it. It depends upon where you are storing your main, disposables, big cartons, and then how you are going to get that into your culture room or adjacent to the rooms. There are three parts. One is the main storage room. Second is the one that you store it for next 8 days. And third is in your actual culture room where you may store it for next day or for 2 days. So these at 3 points, you have to have a complete control of the bacterial contamination or even any kind of contamination. Very often, you just check what is the microbial count at all these 3 places and you will see the difference. You should be able to see the difference. The screening that you will make it on the plates or the agar plates, you just take a part of the dust that is accumulated on your main storage carton and then see what is the bacterial count there. You see what is the bacterial count on your 8 days duration, and then obviously into your drawers where you're going to keep your sleeves open. And then accordingly, you have to maintain the main storage tank to minimize the bacterial contamination.
Okay. And just one quick point here. So Alex, you said you do this every month.
Yeah.
And how often have you found contamination of microbes beyond the threshold?
See that we have seen occasionally the presence of some of the air condition, because in between we had to shut down our AHU, so we were only running with the air conditioner. So in between we had seen the Aspergillus and some of the bacteria like Acinetobacter, more colonies.
And where was this? This was in which city?
Yeah, it is in Kerala only.
So is it safe to say that probably this issue of microbial contamination is heightened in tropical cities with high humidity? And
yeah, definitely that is one of the, and especially like most of the clinics, they don't change the HEPA filter even after 5 to 10 years. Yeah. So HEPA filter itself, I think in one place I saw from the laminar flow hood, during the preparation of the dishes itself, the bacteria were falling. You could see after dish preparation, you could see the bacteria flowing like a Brownian movement. You could see that under the X-ray microscope. So I think one has to be very careful about some of the senders, even the HEPA filters in the laminar flow hood. And you don't know the way they keep it, the vendors.
And when you actually start getting this, microbial, the level starts going up.
Yeah.
Do you start seeing it in culture?
Occasionally we see, like we have seen it in the ICSI dishes. Okay. Occasionally, yeah, because that is one of the dish that is open for a long time.
Okay.
During the manipulations, yeah.
All right. So this is all very important stuff, and for everybody to take note.
Yeah.
All right.
So let's move on to the recommendations. I think we've covered almost everything. So just I put it in the slides. I'm going to just go through the slides. Yeah. I'm not going to speak, but one recommendation from all the panelists and from you, Gaurav, and myself as well. And then we should probably end this. So we can start with Upendra first and then move along. One recommendation that you would want to tell all the attendees here.
Correct. I would talk from filtration point of view, since that is the area I know well. So as we discussed, air quality assessment before and clearly defining your requirements, and proper maintenance of the filters. Testing of filters remaining live at regular interval and changing it. I think that will help us to add into the air quality.
Mr. Jacob?
See, I will quick one suggestion only. I will say there's retrofitting of HVAC systems for a IVF lab in 90% of the cases is not a difficult task.
That's good news for everyone who doesn't have a AHU. It's heartening. Yeah. Now you could expect people who will start calling you at the end of.
No, they will call Alex sir. So in turn, I'll support them. Yeah.
Alex sir is already busy with so many phone calls and emails. I'm very, very certain about it. Vijay sir.
Okay. There are 2 points which I would like to say. One is just analyze and evaluate your air quality at least per your season change. And then second point is just maintain a proper positive pressure into your culture room. These are the 2 points which I can feel that they're important.
Yeah, the 2 points I would like to mention is that there should be a good coordination between the laboratory director or the supervisor and the hospital management, so that any kind of activity goes on the hospital vicinity should be informed immediately, any construction or any renovation. So that kind of coordination is very important. There should not be any unexpected things happen that is the lab is not aware of. Second thing is, I know that it is difficult for many laboratory, but with our own experience, I think many of you will agree. In India, the main problem is with the gases that we get. Compared to maybe all places are not that bad with air quality, but the name of medical gases, I think that need to be taken care. I think that is a valid point.
Alex, you're putting this panel into jeopardy by saying this. But that's a very good points. I will, what my take home from this very enlightening discussion is that we need to continuously monitor. Continuous monitoring of air quality is required, and not only at certain time points. And I personally think that one has to constantly evaluate the effect of the dynamic changes in air quality with the KPIs and the outcomes. So that is very, very important. If we don't do that, then it's a lost cause.
Yeah. Absolutely. And my take home from this panel, and obviously this session today is a custom for patient treatment, and we should look at the lab with the same approach. We can't just offer one solution for all laboratories. It has to be custom made. It has to be tailor made for your location, for your city, for your environment, etc., etc. And all of those factors play a very, very important role in the lab quality that you have in your units. So with this, thank you so much to Ved sir for this kind introduction, and to Gaurav for moderating this. We only know how we've done this, and
but I think guys were excellent. Very good job. The whole presentation. So congratulations on a very good presentation.
And great inputs from all the panelists. I think it's been such an enlightening session for all of us. And again, more power to iHera for all of these sessions.
Thank you very much. Thanks a lot. Thank you. Gaurav, and a good job. Thank you.
Thank you so much, moderators and panelists. For moderators coming with wonderful questions with respect to air quality management, and the panelists coming with excellent answers for the same. So now I would like to invite Dr. Alex sir to deliver a vote of thanks.
Oh my God, that was unexpected. Okay, so it's a great pleasure. It was a good session. I will thank Dr. Ved Prakash to organize this meeting. And also we had a fundamental talk by Dr. Jindal on the basic aspect and quality management, which is a fundamental part of the air quality or the whole IVF scenario, followed by a very stimulating discussion. We had a good team of lab people, plus we had the management team, we had the chemical engineers. So I'm sure that the audience got a good message, practical points that's very essential for many labs across India. So I would like to thank everyone who was involved in this interesting session. Thank you very much.
Thank you.
Good night.
Bye. Take care.
Thanks to everyone. And please, I want to announce one more webinar on 11th November on very important subject, temperature and pH in lab. So please do join on that 11th November. Thank you all of you. Thanks.
Thank you to MCure and Amit as well.
And thanks.
Thank you, sir. Thank you. So from MCure, I thanks everybody. Ved Prakash sir, you has always been very kind to give us this opportunity and will always be there. We are just a phone call away. So thank you, Alex sir. Thank you, Keshav sir. Keshav sir. Thank you, Gaurav sir. Thank you, Upendra sir. You have joined us for the first time. And Sanket sir, Keshav sir, we have been doing a number of webinars, or we have been meeting at number of webinars together. So thank you to everyone for being a part of this.
Thank you so much.
Thank you, Amit.
Night, everyone. Bye-bye.
Good night, everyone. Bye.
So with your permission, sir, I am closing this webinar. Thank you.
Thank you.